How difficult is it to operate a high-content analysis instrument, and is a dedicated resource needed? High-content analysis blends 3 technologies into a single platform. Typically, if you have experience working on a microscope, plate reader, or a flow cytometer, then high-content analysis platforms and software will come pretty naturally to you. High-content tools differ from these technologies due to their image processing algorithms. These algorithms are like instructions and rules that measure phenotypic changes among cells. You should have no problem adapting to high-content analysis if you follow this simple rule. When learning how to use the platform, begin with a simple experiment where you already know the outcome. The platform should report parameters you are used to seeing with other technologies. And what's more, you can test the instrument before you buy and see just how intuitive the technology is. We're sure it'll soon become one of the most powerful tools in your lab. Check out our other videos on frequently asked questions about high-content analysis.